Dear friends, good morning to all. So I got opportunity to present a lecture on the basics of the flow cytometer while we evaluate the semen. So on the demand of the our friends and students, kindly share this video with explanation. So today I am recording this lecture so that you will take advantage, especially students. So we presented this lecture on 29th July, and in this photograph you can see the Joint Director Research and DRI Karnal, Honorable Dr. Rajan Sharma, me and Dr. Megha Pandey from CIRC Meerut. These are the some students of the NDRI Karnal. So we will broadly divided this chapter into four. First, we will discuss the principle and component of the flow cytometer. Then we will discuss what is fluorophore. Then we give some example how sperm samples are analyzed in the flow cytometer. Then finally, we will discuss when you are with your data, how can you present the flow cytometer peaks and data in your research article so that you get published in the peer-reviewed journal. So the basic principle and component of the flow cytometer is, first we will see the definition. The definition of flow cytometer is the word itself explains the definition. Metry means measurement, cyto means cells, flow means fluid. That is, flow cytometer is the measurement of cells in a fluid is called flow cytometer. So basic principle of the flow cytometer is to passage a cells, whatever the cells. It may be sperm cells, it may any somatic cells, it may granulosa cells, luteals, any cells can be passed in a single file. Single file means in a one by one in front of laser. Laser why? So it illuminates the night so that it can be detected, counted, and sorted. So this is the basic principle of the flow cytometer to analyze any cells. So mainly the component of the flow cytometer is made of three main systems. First is the optical systems, then fluidic and electronic systems. So optics system is consist of lasers, mirrors, and filters. So there are three things we have tried to clear it, try to make understand you that what is laser, what is the use of laser, what is the use of mirror, what is the use of the filters. It is very essential to understand it, to know the basic of the flow cytometer, and it will help to select you the different filter, different laser, different mirror for your analysis of your sample. So first of laser, this is the acronym of light amplification by stimulated emission of radiation. So most commonly in the benchtop flow cytometers, there are three lasers are commonly used. One is of color violet, blue, and red. As we know that our eye can detect about 400 nanometer wavelength to 700 nanometer wavelength of light. So these are the in our visible light range. So violet, its wavelength is 405 nanometer, while for blue it is 488, and for red it is 633 nanometer wavelength of the red light. So now how laser is produced in the flow cytometers? The source of the laser is argon ion lasers or solid state diodes. Earlier, these gas lasers are used, but nowadays, to make compact the system, solid state diodes are the source of the lasers. Now, we will try to understand what are the lenses in the flow cytometer and what is its functions. So, you can see in the figure that laser light, one blue laser is striking to a cells, and after striking, some lights are reflected at the 180-degree and some are 190-degree. So at 90-degree, there is a one arrangement of the mirror, which is called collection lens. The function of the collection lens is to collect the interested light of wavelength that we require and avoid other irrelevant lights. Then, after the collection of the light, it goes to the collimation lens. Collimation lens, what is collimation lens? Its function is to make parallel of the light to each other so that each wavelength of light do not overlap to each other. So this is the function of lenses in the flow cytometer, and another light scattered in the at 180-degree centigrade, it is collected by another forward scatter detection lens. So this is about the lenses used in the flow cytometers to collect the light and make it parallel to each other. Now, what is the use of mirrors? Different mirrors are used in the flow cytometer. So once the light, as we have learned that in the lenses, the light has been collected and collimated. Then we have to separate, because once the laser light strike to the cells, it emits different lights of different wavelengths. So each light should be separated for the detector so that a particular light will be detected for the particular biomarkers. So there is a dichroic mirrors, are different dichroic mirrors of different wavelengths are placed at 45-degree angles. You can say it is at 45-degree angles of the light source. So now what is the function? Its function is to pass certain wavelength of light and reflect certain wavelength of light. So there are two types of this mirror, is the long pass and the short pass. The function of the long pass is to pass the wavelength of light which is longer than the certain wavelengths. For example, these mirrors have printed 560 nanometer wavelength, and it is dichroic mirror and it is long pass. It means this mirror pass the wavelength of light longer than 560 and reflect the light 560 nanometer or less than 560. So for example, it passes the light greater than wavelength of 560, it is 600, 640 like that, and reflected less than light of wavelength 495. Here you can see another mirror is arranged to further separate the light. So in this way, different wavelength of lights are separated before the filters and detectors. So that precisely our detector can detect the light of optimum intensity. So this is about the mirrors. Now, what is filter and what is the use of the filter? So after the separation of light by the mirror, it comes to the filter. So in black shadow, it is filter, and gray color, it is the mirror. It is arranged like this one. You can see in this picture. Also, you can see in this picture how after the reflecting the light pass through different mirrors and the different filters before the detector. So there are three types of detectors used in the flow cytometer. One is the long pass, second is the short pass, third one is the band pass. So its name itself indicates long pass means this filter will pass the wavelength of certain wavelength and and absorb or reflect the other wavelength. For example, this filter in indicated by LP 500. It means it is long pass filter, and and this indicated wavelength is 500 nanometer. Means this filter will pass the 500 or more than 500 wavelength of the filters. So and reflect the less than 500 nanometer wavelength of light. Similarly, short pass. Here it is SP, it is short pass. Wavelength uh, demarcated here is the 500 nanometer. It means it is short pass. It means the 500 or less than 500 wavelength will pass and rest will be reflected. So, and third one is the band pass. In band pass, you will find it is written the wavelength. For example, here it is written 500 oblique 50. It means you have to carefully listen. It means it passed the certain range of the wavelengths, and out of the range it will be reflected. So for example, this is 500 oblique 50. It means it is plus minus 25, not it is plus minus 50. Means the wavelength range of this filter is 475 to 455. Kindly note that it is not 500 to 550. So this filter only passes the wavelength ranging from 475 to 525. So this is about the different filters and their functions. So these filters are arranged like this band pass filters in the flow cytometers. And finally, after the filter, this separated light or desired light go to the detectors. And this detector is called photomultiplier tubes. So because we detect the light or fluorescence from a cell, so the luminescence or its intensity is very big. So the function of the photomultiplier tube is to amplify the signals to generate voltage pulse. This voltage pulse is finally converted into digital readout, means electronics, that will be appear on our computer screen at different form a dot, 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 dot that you see in the result of the flow cytometers. And second system is the fluidic system, in which the sample is sucked by the flow cytometers and forced in a single file. For this single file, there is a pressure, certain pressure of this sample, and there is a surrounding sheath fluid. If the pressure is not optimum, this fluid is not, these cells are not come one by one. So there should be optimum sample pressure and fluidic pressure. Generally, the sample pressure is slightly lower than the sheath fluid pressure, so that all the sample come in front of laser one by one. So it will be detected efficiently. So as we know that once the light is strike to any object or any cell here, lights are reflected in different direction. Some light are forward, some light are in the side scatters. So suppose, and some light are absorbed. This is the light source. It is a strike to the cells. Some light are absorbed, some light are reflected inside, and some are forward direction. So those light are reflected in the forward direction. It measures the size of the cells. Suppose if the light is reflected, if size will be this one small, the length between the two forward scattered light will be short. Then that means it is short light. This is very good example of our blood cells, white blood cells, in which we can detect the different size of the blood cells in with the help of the flow cytometers. So this is called forward scatter. Means forward scatter measure or estimate the size of a cell. Now you have to remember this forward scatter for size of a, to estimate the size of a cell. And next is the side scatter. So, as we already told that some lights are absorbed, some are reflected. The reflected light in the side wise reflect, estimate the granularity, heterogeneity, or the refractive index of the cells. Means side scatter detector detect the granularity of the cells. For example, this size of the nucleus is large in this cell. So side scattered will be more, means granularity is more. If less, the side scatter will be less. So it will be detected by the side scatter detector is less. So means it is less complex, less granular. So in this way, side scatter measure the granularity of a cell. Forward scatter estimate the size. Remember it. Forward, size, side, remember, measure granularity. So in this base, on the basis of this forward and side scatter, we select the desired cell population of our interest. For example, sperm. So in sperm sample, when we collect, the sperm sample will be contaminated with the many epithelial cells. Some cells become doublet, or some debris will be there due to the our this extender. So we can distinguish our sperm population from the other cells. This is the function and use of the forward scatter and side scatter. Now we will learn about the fluorophore. So what is the fluorophore? Fluorescence. As we know that light is a form of electromagnetic energy. it travels in a wave. These waves have both frequency and wavelength. And wavelength of the any light determine its color. So for the 400 nanometer, it is the violet, and 700 nanometer, it is red. Beyond this, infrared. Beyond lower than this, ultraviolet. Because you see, lesser than violet, then it is called. That's why it is called ultraviolet. It is more than 700 nanometer infrared. So our eye detect only about 400 nanometer to 700 nanometer wavelength of the light. So molecules that are capable of fluorescence means that can imitate the colorful light is called fluorophores. When a fluorophore absorbs light, as we earlier told that when a light is a strike to the cells or object or anything, some lights are forward scattered, some light are scattered inside, some are absorbed. So some absorbed light, some certain wavelength of absorbed light can excite the electron of the cells or the object. So the electron from the ground state, because light has certain wavelength, light have its own energy. This energy excites the electron of the fluorophores from ground state to excited state. As we know that excited state is unstable, so again the electron will return to their ground state. Here you can say the certain energy excite this electron to a excited state. Again, after some times, it comes to the ground state. Once again the drop to the ground state, energy is emitted and fluorescent light will emission occurs. So here we learn that how the fluorescent is excited by certain wavelength of light. All light cannot excite the electron of certain wavelength, electron of the certain fluorescence to make excite or to emit fluorescent color. So it is a specific for the fluorescence, fluorophore, what light wavelength range can excite. So what is conventional and fluorescence? In this figure, you can see this is regular yellowing and this is fluorescing. So obviously, we see the color of light that are reflected. So in this, you can see the reflection of light is less. In fluorescing, the reflected light is more. So it is called fluorescenting. So fluorescence are brighter, bolder, and better than regular. So in the semen analysis, the most common fluorophores we use are the FITC. We see in the green filter, PI, red filter, DAP, host, flu, mitosox. These are some example of the fluorescence fluorophores that we use for the analysis of different parameters of the semen samples. Here we give an example so that you can understand what laser should be used, what filter will be used, and how can we analyze the sample. So suppose we select one fluorophore is called mitosox. The mitosox, the word is made from mito and sox. Mito here it means mitochondria, sox means superoxide. This fluorophores when incubated with the cell, here a sperm cell, it penetrates the mitochondria. And we know that mitochondria is the double-edged sword. Once the mitochondria produce ATP as well as free radicals. If the more free radicals are produced, it is harmful for the cells. So this main primary free radicals of the mitochondria, and it is only produced by the mitochondria, is the superoxide. That means the superoxide is very specific for the mitochondria. So for the assessment of the mitochondrial health, we estimate the superoxide production of the mitochondria of a cell. If it is less, means the cells have good health. If the production is more, it means the health of the cells is not good. So once this fluorophore attached to this superoxide, it and exhibit the red fluorescence. So first select taking the any fluorophores, we should know about the excitation and about its emission wavelength. So in this example you can see the this is the blue color, the excitation wavelength range. Means this mitosox electron can be excited by the wavelength ranging from 350 to about 600. Means the wavelength of any light ranging from 350 to 600 nanometer can excite the electron of the mitosox from ground state to excited state. After the excitation, this will again come to the ground state and emit the fluorescence. So this is the concept about the excitation wavelength. But here you can see the maximum wavelength intensity, relative intensity, means about 500 when we use, instead of this wavelength 350 to 600, we use single wavelength of about 500 nanometer that will excite the electron maximum. This is called emission excitation peak. Means at when we use the 500 nanometer wavelength, the excitation will be maximum. So this is called excitation peak of the mitosox. So you can say the excitation peak of the mitosox is about 500 nanometer. Means maximum electron will excited with the use of this wavelength. So what will happen? After excitation, emission will happen. So this is the emission wavelength. This color is emission, means it ranging from about 500 to 750 nanometer wavelength. Means after the excitation of the mitosox, it emits the fluorescent light of wavelength ranging from 500 to 750 nanometer. Is it clear? So many color will be emitted by this fluorophore. But again, you send the peak light, means maximum intensity of the light of wavelength around 600 nanometer. You can see here less than the 600 nanometer emitted light will have maximum intensity, means the maximum luminescence, maximum brighter light, you can say. So we focus when we selecting the emission and excitation, we focus the peak. So in this example, you can see the excitation peak of this mitosox is 500 nanometer and emission peak is the 582 nanometer. So this is clear from this spectrum, this graph. But it doesn't mean it cannot be excited by lesser or greater than this wavelength. But this is peak. So now, as we learn that this laser, so there are three lasers commonly used in the flow cytometer. One is the violet, second one is green, and third one is the red. Green one. So for excitation, laser is required. Laser light is required. And for mitosox, the excitation peak is 500 nanometer. So we will select the excitation wavelength of the light so that maximum electron will be excited to emit the maximum energy or maximum light intensity. So we will select the laser wavelength for the detection of the mitosox of around 500 nanometers. So we have this wavelength of lasers are available in our flow cytometer. So for each wavelength, different laser cannot be installed in the flow cytometer. So we have select near to this 500 nanometer laser. So near to the 500 is the 400 nanometer. So this mitosox fluorophore will be excited maximally by the wavelength of the blue lasers of wavelength 488 nanometers. So this is how we select on the basis of this excitation wavelength spectrum. Now emission. As we know that emission peak is 582 nanometers. So which filter we have to detect? Select those are near to this. It means these are the filter available with you with the blue lasers. Blue lasers have arranged 5 filters and you have to select this one filter for detection of the mitosox which is near to the 582 nanometers. So for this green, it ranges, here it is written 525/40, means it is not plus minus 40, it is plus minus 20. So it can pass the wavelength of range 505 to 545 to the detector for detecting. So it is not around this. Now second filter is this color having 585/42 band pass. So it is 582 plus minus 21. It is 562, 606. Means it is come this range, this excitation peak come to this range. It means we can detect this mitosox from the second filter. Now come to the third filter. It is 610 plus minus 10, 600 to 620. It is also you can detect in this filter. So in this filter you can detect both filter, you will detect the mitosox. It can be efficiently detected, but it also detected another wavelength because maximum is 750 and this comes, it will be detected, but the light intensity of we detect the microscope, this filter will be very weak. Signal will be very weak. So we will not select this one. Again, here will be, again can be select. You can see the spectrum, emission spectrum is the 500 to 730. So it also comes to the range. But again, we will use this filter. The emitted light will be weak signal, not a strong light. So we will select these two filter for maximum intensity. So from this slide, we can know how can we select the appropriate excitation laser and the filter to detect the your biomarker present in the your sperm cells. So for example, for any flow cytometric analysis, first we run the sample without the fluorescence to detect the autofluorescence of the cells. So for example, we run the sample without mitosis. So our cell population comes here, forward scattered, side scattered, measure the size and the granularity of the cells. So on the basis of, we selected this population. This may be the doublet or other cells. So we selected this population of, and considering this will be the sperm cells. So this is the all event, and this is here we run 10,000 samples. 10,000 event means we analyze the 10,000 cells. Here 10,000 sperm cells. Out of this 80% we selected. This is called P1 population. Out of 10,000, 80% cells are selected for analysis. So this is the selected population. This is called P1. And when we see the histogram and we autoline segment, we estimated this is the autofluorescence. This is the PE. This is the filter. And this is the intensity of light, fluorescent, X and Y axis. So without stain, the autofluorescent intensity comes to around 10 to the power, more than 10 to the power 3. Means those cells fall, this cell intensity will be considered as no stain. Then we will run the sample incubated with the mitosocs, and we select the P1 population similarly as previous slide, and we see this cell population as in control. It was the autofluorescence, means this fluorescence intensity of light is come due to the autofluorescence, and this intensity of fluorescent light is due to the mitosocs. Means these cell populations are mitosocs positive and this is negative. And when we see the autoline segment, we can see the out of this P1 population, 58% cell population was mitosocs negative, means there is no excess production of the superoxide in the cell population, while 41% cell population have mitosocs positive, means mitochondria produced excess superoxide. So these mitochondria is not healthy. Ultimately, your cells are not healthy. So there is a option for a state. So we can estimate here statistics. So all event 10,000, 100%, P1 74%, P2 41%, P3 58%. P2 is the mitosocs positive, here P2, and here is the P3 population. So in this way, we can know the the status of the superoxide production of our sperm cells. So we counted 10,000. We can also count 20,000. So in fluorescent microscope, you count 2,000 sperm cells. Here we counted in 1,000, 10,000, 20,000, 40,000. So efficiency and reliability is more for the flow cytometer than the fluorescent microscope. We regularly measure. Our Semen lab is accredited by this central monitoring unit, and we are regularly monitoring the mitosucks production by the our cells in the both three conditions: fresh semen, equilibrated after 4 hours at 4 degrees centigrade, and after freezing. So we have a thousand data on fresh, equilibrated, and frozen. And we can see here the remark, those ejaculate have been discarded. If you see carefully in the phrase, this ejaculate of bull 191 having the mitosucks in the range, but it is discarded. And this ejaculate of bulls 5511 having initially very high concentration of the superoxides, and both are discarded. In frozen, it is again increased. So we observe that in the phrase, 5 to 50% sperm cell are mitosucks positives. In equilibrated, it is 50 to 30%, and in frozen condition, maximum 50% sperm population are found mitosucks positive, means mitochondria of the sperm cells are producing more superoxides. So in this way, you can estimate, you can know the sperm cells health. Now, second example, we will see the acrosome integrity. Normally, by Giemsa stain, we stain and we know the the sperm's acrosome are intact or damaged. But fluorescence also we can observe. This fluorescence, when attached with the PSA or PNA, this PSA and PNA penetrate the acrosome. If the acrosome enzymes in the are present in the sperm head, if it is leaked, there is no enzymes, so PSA will be no inside the cells. The fluorescence will be low. This is the basic principle of the know the acrosome intactness of the sperm cells. PI stain we add to know those sperm cells are intact, but whether it is live or dead. If it is dead, instead of their acrosome is intact, it is no use of ours. It is not useful for the fertilizations. So if you use the two fluorescence FITC conjugated with the PSA and PI, 4 combination can be made. Live sperm having intact acrosome, live sperm, but their acrosome is damaged. Dead sperm, but their acrosome is intact. Dead sperm and acrosome is also damaged. So for our interest is the live sperm and intact acrosome, means sperm both acrosome should be intact and sperm should be live. This is useful. So we will estimate this live sperm and intact. Live sperm means PI negative. Intact acrosome means FITC positive. So you should remember, if PI penetrate the cells attached to the nucleus, means the cells are dead. So PI negative means dead cell. PI negative means live cell. PI positive means dead. Opposite is that FITC positive means intact acrosome, FITC negative means damaged acrosome. So similarly, first we run this semen sample without S10. We select the P1 population and we divide it into quadrants. So this is the autofluorescence because we have not this. This is the light intensity. This is the FITC. FITC and this is PE filters. We use here FITC and PE filters on the basis of these two fluorophores, excitation and emission wavelength, mainly emission wavelengths. So this is the fluorophore. Here, this negative run of the sample indicate this is negative for FPI, negative for FITC. Means if the sperm population will fall in this quadrant, means this is FITC. This for FITC, this is the borderline. Means above this, no, sorry, just this line, this vertical line indicate if sperm population fall in this quadrant, means FITC negative. Beyond this, FITC positive. For PI, means live dead. This line indicate the borderline, means below this cells are live because it is PI negative. And above this line, the sperm population falls. It is PI positive, means it is the dead cells. Now we will run the fluorescent samples. We incubated the sperm sample with the FITC and PI and run the sample. We found the different cell population in the different quadrant. This is but this is not clear-cut divided groups. They are mixing, overlapping to each other. So why this happen? Otherwise, our result will be biased. So we will now, what is compensation? We will learn about the compensations. Here we example two fluorophores, FITC and PI. So this is the excitation wavelength and the emission wavelength. We will focus on the emissions because both the wavelengths can be excited by the 488 nanometer due to its peak excitation peak. Here also it is you will see the excitation peak is around 500 and similarly for the PI it is around 500. So this will be excited by out of three. Both the fluorophore will be excited by the blue laser of wavelength 488 nanometer. So if for the emission, it is very important for this. For the emission wavelength of the FITC ranging from 500 to 650. Remember it. 500, about less than 500 to about 650. Peak is about 525 here 525. Emission 475, 650. Similarly for PI, its emission spectra is the 535 to 725 and maximum is at 600, 180. Here maximum 525 and 680. So earlier I told that we decide the filters, what should we use on the basis of the peak emission. So on the basis of the peak emission, we will select the filter which wavelength is around 525 nanometers. So its wavelength is the 525 nanometers. So for FITC, most suitable filter will be this green one. Is it clear? Now, for the PI, maximum wavelength is 618. So it will come this range, will be detected by this yellow one, 585/42. So FITC will be detected by the blue filter, and also it is called FITC filter, and this is called P filter, or this is P filter, or this 585/42 band pass. Clear? But here is the twist. Earlier I told that it is not necessary, it is only detected by this one detector. Here, the wavelength ranges 300 to 550. 550, 550 nanometer. So, this 585 plus minus 42 also detect the FITC. Think, listen carefully. How it is possible? Once this 550, we will minus 21. So it will come to the range of this. Here in the emission spectra is around 650 emission 650. So it will comes to this range when you add 585 plus 21. So this wavelength 650 may be detected by this one. So FITC not only detected by so emission spectra of FITC ranging from 575 to 650 means this spectra, this FITC can be detected by this one, this one, this one, this one. Means four filter can detect the FITC. However, the maximum detection or maximum intensity of light detected by the green one. That's why we use the filter to detect the FITC 525/40 bandpass filter or green filter. And we observe the color of the FITC will be the green. But it is not like that. It can be detected by this one filter, this one, this one. So similarly, PI, the spectrum, emission spectrum is ranging from 535 to 725. But peak is 680. That's why it is mostly detected by the PE. We detect in this filter, but it can be detected in this one because it's the lower side wavelength range is 535. When we plus the 525 plus 20, it is 545. So it comes to this range. Means green filter or FITC filter can also detect the PI. So here is the overlapping of the wavelength of emission of FITC and PI. So in this figure, you can see this is the first filter, this is one second filter, this is 525 and this is 585 filters. So maximum emission of the light is in this 525 of FITC, but when you see the graph of the green comes to this range 585 to 85 here, the red light, this red color means this cell population can be detected by the PE filter or PI filters. Means the cell, suppose the PI positive cells are 70% actual and 10% come for the FITC positive cells. So detector will detect 70 plus 10% of the FITC positive because the cells was PI positive means dead and FITC means acrosome intact. It will be also detected by these filters means 70 plus 10, 80%. So it gives false positive result. So therefore, similarly, when see the spectrum, emission spectrum of the PI, it falls in this filter also, this blue light you can see. Means again, PI positive cells will be detected by the FITC positive cells. So again, you will get the FITC positive cells, positive, false positive results. So we have to separate this. So we can compensate, means we can do, we can replace back and this filter this back so that the wavelength of light do not fall to one fluorophore nor to the other filters. So this is called compensation. So how can we compensate? See, for example, this is the cells which is FITC positive. This cells, this shadow indicate the in this range the cell falls are FITC positive. This is FITC negative cells. Again, then we add PE only in these cells. We incubated with FITC. In this cell population, we incubated the PE. This is the PE positive cells and this is PE negative cells. When incubated the cell with PE and FITC, this wavelength, the positive cell will be overlapped. You can see. So how can we do? We drag this cell population to here, our screen with the help of the software. And then we see the cell population are separated when we run both FITC and PE. Similarly, every bolts will be measured for the acrosome integrity every month with the help of flow cytometer, and we found that in fresh semen, the ejaculate past the PTM result ranging from 70 to 90% in fresh condition, and in incubated it is around 60 to 80, and in frozen condition it is ranges from the 40 to 70. This sample ejaculate were discarded. So you can see the live acrosome intact, means cells was live and acrosome was intact, was only 23 and it is only 17. So it is discarded on the basis of PTM. Also, it was poor in the acrosome integrity on the basis of the analysis by the flow cytometers. Now, similarly, we measure the mitochondrial membrane potential. Here we use the dye JC1. So when the mitochondrial membrane potential is high, after penetrating this JC1 aggregate inside the mitochondria and emit red light, another wavelength of light. When it is in monomer condition, emit another wavelength of lights. So this aggregation and monomer and emission of light, we can distinguish the high mitochondrial potential, low mitochondrial potential. So for example, this is the wavelength spectrum of this monomer and aggregate. This is the excitation, and you can see the excitation wavelength of monomer JC1 and aggregate JC1 is almost same, but emission wavelength is different. Here you can see this is this is monomer. It ranges from 500 nanometer to 610. It is 500 to 610, and aggregate emission spectrum ranging from 550 to 630. So again, this emission wavelength is overlap, means it will be detected, and peak is for the monomer is 530. If peak is 530, automatically we will select this filter. It is around the 525 plus minus 20. And for aggregate, it is around 600, 585. So we will detect this aggregate in the second filter, PE filter, or this 585 oblique this filter. So we will detect this one, but the emission spectra of the monomer and aggregate is overlap. So we have to apply the compensation, otherwise we will get the false positive result. So it is very important to apply the compensation where the emission spectra of the two fluorophores are overlapped. For this, you have to know not only the peak, but its spectrum range is essential to know to apply the compensation. So, as usual, first we will run the sample without the stain. So, sperm population all here, and this is not, we apply this. So monomer will be detected by the first filter, FIT, it is called FITC filter also, and PE will be detected. This one means if the population having the intensity of the light, intensity will be higher, and from this boundary, it will be the low mitochondrial potential, and from this, for the PE, it will be higher, it will be high mitochondrial population cells. Once they run the stain sample, you will find there is no separation of these two groups of the population, high mitochondrial sperm having high mitochondrial potential, sperm having low mitochondrial potential. So again, we have to apply the compensation. We apply the compensation, we find the two population is almost separated. This group is high mitochondrial potential. This is the low mitochondrial population. So in this way, in another publication, it is not my publication, we can say how they have separated the two population of monomer and aggregate. So initially it was this one. So apply 10% compensation, 20%, 25%, and 30% compensation. In 30%, clear cut do two population, high mitochondrial population and low mitochondrial membrane potential separated. So we regularly measure again, the mitochondrial membrane potential in the phase. It should ranges from 85 to 95 in the equilibrated condition. It ranges from 70 to 9, and the frozen it is 50 to 60%. For the that ejaculate qualify for the incubation test, PTM test. So most important thing is the maintenance of the flow cytometer. Many research institute procure the flow cytometer, but they are not able to maintain. So ultimately they are dumped. So very critical to maintain the flow cytometer. For this, whether you are performing experiment or not, you have to clean the sample every 2 or 3 days. And every week or fortnightly, you have to clean with deep clean solutions. So whether you are running the sample or not, you have to clean it regularly and quality control. So before the run the sample, there is quality control beads, QC beads. It is called QC beads. So you have to run QC beads. Instead of cells, we run beads. Have predetermined number of the cells. So can we detect all the filters are working properly. So if it is not working, there is no use of run the sample. It will give biased result or false result. So first we run the QC bead, and when it is passed, it come tick, 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 green colors. When it is not passed, it will come red colors. And another important, you have to maintain the flow cytometer if you have a contract for annual maintenance. Otherwise, this will not work smoothly, and you have to change some consumables, pipes, and regularly at every years. For this, you have to purchase PM kit, means preventive maintenance kit. Then and then you can maintain your flow cytometer. You can get the accurate result. Otherwise, no use to procure the flow cytometer. So now last chapter is how to publish your data in the peer review research articles. It is very tricky also. It depends on your data. How can you, how much you have data, and also figure legend. You have to, everything you have to write. Otherwise, reviewer will not accept. For example, we have write flow cytometer getting to perform how much we have counted 10,000 cells, which laser we have used, which filter we have used, how we have getted auto line segment, and how it is, means we have to clearly everything you have to mention in your figure again. Just if you write, we have measured this mitosocs with the help of flow cytometer, so it will not published in peer review journals. So now what picture we have to include pic of the flow cytometer? It depends on your data and the journals. If you very mediocre journals ranging from 1 impact to 2 impact factor, where and you have a few data, then you can put all the steps. For example, the forward scatter, side scatter, selection of population, positive control, negative control. We have run the positive sample, then the negative sample, then the bar diagram. Many times there is a restriction of the figures. In that condition, you cannot put all the figures of the flow cytometer in your publication. So just for the flow calcium indicator, we just put one last this quadrant in which flow positive, flow negatives, PI positive, PI negatives, and there this bar diagram. So it depends on the data and how many figures of the journals are allowing to put in the manuscripts. Similarly, we published in the scientific report only we for this data, and rest of the data we provided in the supplementary figures and tables. So this way we can analyze the samples. This is our semen freezing lab, and this is the ICR first semen freezing lab accredited by Central Monitoring Units. He is my director, Worthy Director T.K. Dattai, Dr. R.K. Sharma. He is overall in charge, myself, Dr. Pradeep Kumar, and she is the Renu Bala. She is the maintaining our flow cytometer, run the sample from everywhere comes. It comes from NDRI, Gadwasu, Luvas, everywhere. We are welcome all the samples without any hesitations. We run all the samples if it is possible for me. For your kind information, we have developed and the technology developed. We developed digital AV for semen collection, a spermoscope to analyze sperm motility in the field condition. And we have also developed a ready-to-use semen extender. It is egg yolk free. And recently we have filed the patent. We have developed a unique CIRB AI gun. Thank you.